All right, let's start by going over the genetics packet page 1 items. You'll want to have your packet out and you're going to want to fill in items as we go over them. You'll want to hit pause from time to time on the video to let yourself obviously catch up and what have you, and here we go.
All righty, well, welcome to the new version of school in the Gerby household. This is what it looks like when you're doing it Corona style. All right, so first thing we have to talk about, characteristics versus traits. Pretty much similar in the regard, but there is a slight differential, and here's what it is. Characteristics. Characteristics are going to be things that are more overarching. So what do I mean by that? I've got a couple examples here, okay? Earlobe attachment. So if you looked at your earlobes are going to be one of two things. They're either going to be free. And what does that mean? It means if you looked at where your earlobe meets your head, there's a little bit of a separation. I have free earlobes. But some people, their earlobe just attaches directly to their head. And we call that attached. This is an example, or these are an example, of a trait. It's just more specific. Free earlobes, attached earlobes. All right, hair texture, let's give you a couple of those. So the characteristic is overarching. You have some kind of texture to your hair. But there are some options, right? You could have straight hair. And that is a trait. But you could also have curly hair, right? And curly hair would be a trait as well for a characteristic, which is hair texture. Lastly, let's give you another example of a characteristic, okay? Eye color is the overarching characteristic. However, more specifically, you might go on to say the person has blue eyes. That'd be a trait. Somebody has green eyes. Somebody has brown eyes, etc., etc. And so just the big difference between characteristic and trait is characteristics are going to be more overarching, more general, whereas traits are going to be more specific. Alrighty, it's on now to Mendel's Principles. So there are three primary principles I'm going to teach you about right now, okay? They're on your packet. And they are the law of segregation, the law of independent assortment, and the law of dominant and recessiveness. So let's talk about the law of segregation first. And by definition, what it says is that an organism can only pass one allele for each trait to an organism. Okay? So let me give you an example, okay? So you're going to hear about something called homozygous and heterozygous going forward. But let's say for, I don't know, pea plants, okay, that a pea plant is big T for tall and little t for short, okay? That's its genome. I got one from its mom, one from its dad. When it goes to have a baby pea plant, right, it can only give either big T for tall or little t to a pea plant, but it can't give both. So that's the law of segregation. One allele for each trait. Now, law of independent assortment says this, that these genes separate independently from one another. Okay? And so whenever I talk about this, I always think of my high school homecoming date, okay? Her name was Janelle. Now, a lot of people think, so they separate independently from each other. So a lot of people think that, you know, if you have dark features, right? So if you have like dark hair, well then you must have dark eyes, right? Well, you know, my homecoming date, Janelle, had dark hair, darker complexion on her skin, and just bright blue eyes. Because the law of independent assortment says, you know, your hair color, your eye color, etc., are all independent of one another. Now, do those things often end up together? Yes, because overall in the gene pool, dark features tend to be coupled with dark features. But it's not necessarily a has to happen thing. And then the last thing, the law of dominant and recessive. I'm going to go back to this big T, little t, because this is a pretty simple thing to teach. So what ends up happening is this. The law of dominant and recessive says that a dominant allele masks the recessive. So dominant masks recessive in a heterozygous relationship. So in heterozygous. And again, what does that mean? It means that if you have a big T and a little t, the little t won't express, all right? Because there is a dominant T present. So there are two ways you can be tall if you're a pea plant, right? Big T, right? The dominant or the dominant. But the only way to be a short or a smaller one would be to have a little t and a little t, so there was nothing dominant with the dominant feature to mask this recessive one. So those are Mendel's principles. All right, next let's cover generations. So you're going to see P, F1, and F2. So what do they mean? Well, P stands for the parent generation, okay? F1 means filial one, okay? Filial one. What does that really mean in layman's terms? It means the first generation. And F2, what does that mean? It means filial number 2. So I'm going to do a little bit of pre-teaching here. The next topic we're going to talk about is something called Punnett squares, okay? So let's say we had two parents, right? And again, we'll use tall. So big T equals tall in pea plants. So let's say we had a homozygous tall parent, big T, homozygous meaning those two letters are the same, crossed with a homozygous short. In fact, and that's the only way to be short, right? Otherwise the T would have masked it, like we just talked about. And so what ends up happening here is, no matter what in this thing, right, Punnett squares. So if we draw a Punnett square here, all right, now there are 2 letters here and 2 letters here. That's why our Punnett square can have 4 squares, 2 times 2. And so what would happen is, right, law of segregation would say this parent has to have a big T or give a big T. And so this one is the left one, this one is the right one. And the same thing would have to happen across the top, right? We'd have a little t and a little t. And so what would end up happening is we do this like a matrix. We bring them down and over. So big T, little t, big T, little t, big T, little t. So they're all genotypically big T, little t, and phenotypically tall, which we'll talk about that in a future video. So filial generation 1 would say if we took 2 of these and crossed them together, so we had a heterozygous now, crossed with a heterozygous, right? Well, 2 times 2 letters, but it's a little different on this Punnett square, all right? What ends up happening here, okay, is we end up having big T half the time, but half the time would be a little t. Well, this organism, half the time big T, half the time little t. So what would we end up with? We'd end up with big T, big T, big T, little t, big T, little t, and little t, little t. And so we'd have 1 big T, 2 big T, little t's, and 1 little t, little t. So we'd have a 1 to 2 to 1 genotypic ratio. Our phenotypic, what it looks like, would be tall, 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 and short. So we'd have 3 to 1. And then likewise, if we were going to keep going, which I'm not, we would then take 2 of these. We could just pick any of them. We'd cross them again to have another generation, and so on and so forth. But that's what P, F1, and F2 stands for. All right, the last problem that you see, or the last 2 problems, says how many different types of gametes would an individual of the following genotypes produce? And then it talks about what would those gametes be. So the first one is how many different types, and that's pretty easy, okay? So here's how we do it. Remember, we have this law of segregation that occurs, right? So when we look at these, right, for the very first one, no matter what, this individual is going to give a big A to an individual, whether it's this one 50% of the time or this one 50% of the time. So as such, always going to get a big A. So what are that number of possibilities? Just 1, right? Big A. Same thing here, right? 50%, 50%, but no matter what, getting a big B. So 1 possibility. Same thing here, same thing here. And so how many possibles? Well, we multiply them together. 1 times 1 times 1 times 1. There'd only be 1 possible sperm or egg that could happen. This is a little different here, okay? Big A, little a, so 50% of the time a big A, 50% of the time a little a. That is 2 distinct possibilities, so we note that, 2. Big B, little b, again, 2 distinct possibilities. Big C, little c, 2 possibilities, but for big D, would be a big D no matter what to only one possibility. Again, we take those things, we multiply them together. 2 times 2 times 2 times 1, and that would give us 2, 4, 8 times 1, and that is 8 different possibilities. Here we have 2 possibilities, 2 possibilities, 1 possibility. 2 times 2 times 1 would be equal to 4. Now the last question, okay, says, how do we know what they are? Meaning, what does that sperm or egg actually look like? So let's do this one. We'll break it down. It's going to take us a little more time. So for this first one, big W, we'd only have one possibility no matter what, a big W. Here we'd have 2 possibilities, a big T and a little t. And here we'd have 2 possibilities, a big P and a little p. So 1 times 2 times 2, there are 4 possible gametes, okay? Well, what are those 4 possible gametes? Well, here we go. In order to do this, we have to do something called a factorial tree. So we only have one possibility. So that is cool because when we do a factorial tree, what that means is the only possibility was a big W. Now for the second one, we could have had a big T, but we could have also had a little t. And that's what a factorial tree does, is it makes us follow along. Now, for this big T, it could have had a big P or a little p as well. So there's big P and a little p. And likewise, for this little t, it could have also gone big P and little p. And then what happens is we just trace along what we see now. So across the top, big W, big T, big P, that was one possibility of a sperm or an egg. We could also have big W, big T, little p. We could have big W, little t, big P, which is this one, or big W, little t, and little p. And those are your possibilities for that. And that's gamete combinations.
Now at this time, you should go ahead and complete any items on here that we didn't complete already. That would include the characteristic versus trait items. And yeah, so get those things done, and in a minute, I'll post a picture of the key. So you're going to hit pause right now. And like I promised, here are the sample answers. You want to check those over. Make sure you got them right.